So hello everyone here. I'm Jiacheng. I'm a fifth year PhD student in biomedical data science at UW Medicine. So today I'm going to share with you with our recent work on how to do valid GWAS or machine learning predicted outcomes. First is some background. So GWAS is to assess the associations between the millions of SNPs and the disease or a trait. So the basic idea for GWAS is that you collect a lot of samples, you collect their genotype and the phenotype informations, and then you want to see which genetic variance is associated with your outcome of interest through millions of marginal regression analysis. So here is a typical result from the GWAS. Each dot is a genetic variance, and you have a p-value for each variance, indicating the evidence for that variant to associated with your phenotype of interest. However, for a lot of the valuable outcomes, there is a missing data problem in large genetic data set. For example, this biobank data have been always used in many of the GWAS, but for some outcomes such as X-ray or brain imaging data, there's a very large missing rate. Over 90% of people have this missing outcome measure. And similarly, for gene expression data in the GTEx, a lot of tissue have the missing gene expression data. For example, in the brain, people need to donate that brain to measure the gene expressions. So there's a missing data problem. So if people just do a GWAS on these observed outcomes, then it may give us a lack of statistical power. So to address these problems, in the literature, people have proposed a new study design called machine learning assisted GWAS. So the basic idea here is the data set you collected, here is the phenotype you collected, but if you only run a GWAS here, you will lack statistical powers. So the idea is people want to use this whole data set that with the genotype informations. So they are using this easy to measure covariates and use machine learning to impute the phenotype, and then directly conduct a GWAS on the imputed outcome. So using this study design, people can leverage all the samples in this data set to do a powerful GWAS. And this study design has been extremely popular in recent genetics literatures. So all these papers adopted this study design. And here in our paper, we show that this is actually a pretty bad idea. We show that there are pervasive false positive associations if we are using the current study design for the machine learning assisted GWAS. So here is the real data benchmarking we do. So in the YouTube biobank, we have everyone's type 2 diabetes status measured. We consider that as gold standard. But we masked everyone's type 2 diabetes status, and instead we use machine learning to predict their type 2 diabetes status and do two GWASs, one on the ground truth type 2 diabetes, and the other on the imputed type 2 diabetes. And we found that over 80% of the associations identified in imputed type 2 diabetes cannot be replicated in the ground truth type 2 diabetes. And to explain these pervasive false positive associations, we propose a model to explain the reason. So when we check our machine learning model, we found that the hemoglobin A1C, the HbA1c, is the strongest predictors of the imputed type 2 diabetes. So you can imagine that if we do a GWAS on the imputed type 2 diabetes, we will capture a lot of the signals from the HbA1cs. And the basic idea for the HbA1c, the idea is HbA1c, although it is used as a clinical biomarker for type 2 diabetes, but HbA1c measures how much of the glucose is attached to the blood cells. So you can imagine if some people just simply have a normal control of the glucose, but that people genetically have more blood cells, then that people will have elevated HbA1c levels. So here is our proposed mechanisms for these false positive associations. That is, the SNP is associated with the properties of the blood cells will be identified as the false positive associations in this chunk. And indeed, when we check our top results in our imputed type 2 diabetes, all these, here are 4 SNPs actually, but there are some phone issues. But basic idea is all of these top 4 SNPs are associated with imputed type 2 diabetes and HbA1c, but none of them are associated with the outcome that measures the control of the glucose, such as the fasting glucose, but all of them is associated with the property of the blood cells, such as the blood cell count. So this indicates that if we just ignore the uncertainties in this machine learning models, in these machine learning predicted phenotypes, and consider the GWAS on that imputed phenotypes as gold standard, then it will give us a lot of pervasive false positive associations. So to address these problems, we propose a new statistical framework called post-prediction GWAS that allows us to do valid and powerful machine learning assisted GWAS. So this is the conventional design for machine learning GWAS, machine learning assisted GWAS, and here is our proposed study design. So the key idea here is we need to impute the phenotypes also on the label data that have the gold standard outcome measured and do GWAS there. So the basic idea is we want to compare these two GWASs. One is on the gold standard phenotypes, and the other is on the imputed phenotypes. If these GWAS are the same, this may indicate that if we do an imputed GWAS using a much larger sample, this can be a valid GWAS. But if this GWAS differs a lot, which indicates that we cannot just trust this GWAS, we need to do some sort of de-bias. So here is our proposed estimator. The basic idea is we will use the difference between these two GWAS to de-bias this GWAS solely relies on the imputed phenotype. So here is our key idea. And there are several key features of our framework. First of all, it is assumption lean. For the assumption lean, we mean that you can use any machine learning model you want. You can use deep learning, you can even use large language model to predict the outcome you want to predict. And our framework will always give you a valid control of the temporal error and the confidence intervals. And our method works for any data distributions. And compare with the current baseline that is only using the label data to GWAS, our method will be no less powerful compared with that GWAS, and it will be more powerful with more accurate machine learning predictions in these unlabeled data. And we even proved that a machine learning assisted version of the GWAS small term theorem here. This is saying that if you have all these outcome measured, you cannot beat our method. Our method is the optimal solutions for a valid machine learning assisted GWAS. And furthermore, it only requires summary stats input. So you can imagine that you can use the state-of-the-art GWAS software to produce the summary stats first, and then combine this, do a linear combination of these summary stats. And because of this, it is very fast. It can complete analysis of 10 million genetic variants in 3 minutes. So that's the key feature of our method. And we do some simulations and it shows it works pretty well. So there are some color issues, but the basic idea is if you do a GWAS, here is the red dot, on the imputed phenotype, you will get a lot of false positives. And do a GWAS on labeled, or using only labeled data, and our method does not have any false positives. And with more accurate machine learning model and more unlabeled data, you will achieve more power. And furthermore, we also apply our method in our real data benchmarking example to show that all these associations in the top association with imputed type 2 diabetes is no longer significant in our pop GWAS framework. And all of these are associated with the properties of blood cells instead of the control of the glucose. And so next, we apply our method to the X3 measured bone mineral density, which is the gold standard measurement to assess the fracture risk in the clinic. So compare with the basic ideas in the UK biobank, only 10% of the samples have this DAXA bone mineral density measures. And compare with doing a GWAS only using these 10% samples, we are using 10% of the gold standard measurement plus 90% of the machine learning predictions. So we showed that we can identify 40% more associations compared with only using the 10% data. And we have achieved a very high replication rate. And an interesting result we found is that because there are many skeletal sites that can measure the bone mineral densities, and we found a head-specific signal that is only significant in head, but not in all other body sites. And we found some evidence to show that if we knock out this gene in mice, then it will accompany with some head damage, and the mice were dead. So here are some interesting results. So so far I've talked about how we do GWAS with machine learning predictions, but GWAS is only linear regressions. So we also generalize our pop GWAS framework to answer more broad questions that how can we draw reliable scientific conclusions from black box machine learning model for a wide range of statistical tasks instead of just focus on linear regressions. So here is the imputation-based statistical analysis. That is, you have a pre-trained machine learning model, for example, AlphaFold, and you can use AlphaFold to give you some conclusions, do some statistical inference, give you some conclusions. Or you can have some gold standard measurement of the protein 3D structures, and you can do some statistical analysis there. So here are two common ways people do analysis in the literatures. And here's our proposal that both use the gold standard experimental data and using the large-scale machine learning predictions to give you a machine learning assisted statistical analysis. And next I will show it works pretty well, but our method name is called post-prediction summary stats inference. So the basic idea is, because I've just talked about linear regressions, and we show that we have developed a simple protocol that relies on existing software to only produce the summary stats, and then linear combine these three estimates. And we show that our method can be easily applied to a wide range of statistical tasks, such as quantile regression, instrumental variable regressions, or debias lasso for high-dimensional problems. So our software for both method is publicly available on GitHub, and as you can see, it's pretty simple. 4 lines of code for pop tools and 1 line of code for PSPS. So to summarize, we showed that if we just do a GWAS on the imperfect machine learning predictions, it may give us pervasive false positive associations. And we propose a statistical framework called pop GWAS to ensure reliable genetic discoveries from imperfect machine learning predictions. We also generalize that linear regression to a wide range of statistical tasks. And the paper and software is available. So pop GWAS is just got published in Nature Genetics this Monday. And our PSPS paper is accepted at NeurIPS this year. So I would like to acknowledge all my co-authors and especially my advisor, Chengshi. So that's all I have, and thank you so much.
Yeah, thanks for the overview. So you spent most of the time talking about GWAS and pop GWAS, and I was hoping you could return to PSPS because that was covered a bit more quickly, especially the flow chart on the right side here. Mm-hmm. So could you walk through in a bit more detail, if you have some black box model, I think AlphaFold or structure prediction is the example you gave, how would you use PSPS to perform this assisted inference? What would a practical workflow look like?
Yes. So the basic idea is you have some scientific questions in mind, and you want to use some statistical software to give you some like hypothesis testing to test whether your scientific hypothesis is true or not. That's our goal. And here, if the basic idea is we can use AlphaFold to generate a lot of AI-generated protein structures, and we can use these AI-generated protein structures to do statistical analysis on it to answer our scientific conclusions. But here we are saying that is pretty bad ideas. And you need to generate some sort of the gold standard experimental data to supplement with that statistical analysis that only relies on these AlphaFold predictions to give you valid scientific conclusions. So here, the example is, this is AlphaFold predictions, and then you have some analysis routines that can give you, for example, p-values for the scientific hypothesis you want to testing. But this is invalid, so you need to generate some sort of gold standard data to supplement with these machine learning predicted conclusions, and then give you valid conclusions. That's our basic idea here.
Okay, thanks for walking through that.